The first stage of mitosis is interphase, which accounts for 90% of the whole cycle. Interphase is divided into three subphases, G1, S, and G2. During the G1 phase, the cell grows. In the S phase, the cell keeps growing until it copies its chromosomes. Finally, in the G2 phase, the cell completes the preparations for cell division. In the last subphase, the nuclear envelope encloses the nucleus, which contains at least one nucleoli. Centrosomes are regions in the animal cells that organize the microtubule of the spindle. Two centrosomes are formed during duplicating a single centrosome that contain two centrioles. Centrioles, which are cylindrical organelles that are located near the nucleus and occur in pairs that help with the formation of spindle fibers in cell divisions. Chromosomes that are duplicated during the S phase are not yet condensed. In the second phase of mitosis, which is prophase, the chromatin fibers become tightly coiled and condense into separate chromosomes, and the nucleoli disappears. Each duplicated chromosome is shown as two identical sister chromatids that are joined at their centromeres. The mitotic spindle begins to form in this phase, which is made of centrosomes, and the microtubules extend from it. Radial arrays that extend from the centrosomes of the short microtubules are called asters. The centrosomes are moving away from each other due to the lengthening microtubules in between. The next stage, which is in between prophase and metaphase, is called prometaphase. In this stage, the nuclear envelope starts to fragment and eventually disappears. The microtubules are extending to the point of invading the nuclear area. The chromosomes condense even more. The chromatids of these chromosomes have kinetochores, which is a protein at the centromere, in which the microtubules attach to them and jerk them back and forth. The non-kinetochore microtubules interact with the spindles on the opposite ends of the spindle. In the next stage, called metaphase, the centrosomes are on the opposite poles of the cell. Additionally, the chromosomes are all lined along the metaphase plate, which is a plane that is equidistant between the opposite poles of the spindle. Each microtubule is attached at the kinetochores of these chromosomes. The next stage is anaphase, which is the shortest stage of mitosis that only lasts a few minutes. In this stage, the cohesin proteins are cleaved, allowing the two sister chromatids to part. The two daughter chromosomes move to opposite ends of the cell as the kinetochore microtubules shorten, since they are attached to the centromere. The cell elongates as the non-kinetochore microtubules lengthen. The two ends of the cell have equivalent amount of chromosomes. The second to last stage is telophase, where the two daughter nuclei in the cell start to form, and the nuclear envelope starts to rise from the fragments of the parent cell's nuclear envelope. The nucleoli starts to reappear as well, and the chromosomes become less condensed. The spindle microtubules start to depolymerize, and there are now two genetically identical nuclei. The final stage is cytokinesis, where the cytoplasm is divided. In the animal cells, cytokinesis involves a cleavage furrow, which pinches the cell in two.
This is the process of